Welcome to the Explainer. Today, we're taking a deep dive into the microscopic world to see the very first giant leaps life ever took. We're going from the absolute simplest cells you can imagine to the complex blueprints that gave rise to, well, everything.
So how did that happen? How did we get from a single, simple cell to the incredible diversity of life we see all around us? Well, the answer starts with two crucial kingdoms of life that literally formed the bridge between that ancient, simple world and the complex world of plants, animals, and fungi.
And it all starts right here with Kingdom Monera. These are the prokaryotes. Think of them as the original architects of life. Now, their structure is super simple, but as we're about to see, their impact on the world is anything but. You know, the story of Monera is really a story of two sides. Most bacteria are heterotrophic, which just means they get their energy from other sources. A lot of them are absolutely essential. They're the decomposers, recycling nutrients, or the little engines we use to make things like curd and antibiotics. But then there's the other side of the coin. This kingdom also includes some pretty notorious pathogens, the ones responsible for diseases like cholera and tetanus. Oh, and a key point, even photosynthetic bacteria, like cyanobacteria, belong right here in Monera, not with the plants. And then you have this guy, Mycoplasma. What a fascinating little organism. It's the ultimate rule breaker of the bacterial world. Not only is it the smallest living cell we know of, but it completely lacks a cell wall, which is a defining feature of almost all other bacteria. This unique trait makes it a classic trivia question, and definitely something you'll want to remember. And just to top it off, it's also pathogenic, causing diseases like pleuropneumonia. All right, so from the simple prokaryotes of Monera, let's see how evolution took its next great leap. This brings us to a kingdom that would completely change the game, Kingdom Protista. And this right here is a monumental jump in complexity. Protists were the first organisms to have their genetic material tucked away inside a true nucleus. This was the evolutionary innovation that paved the way for every single advanced life form on this planet. The best way to think of Kingdom Protista is like nature's grand laboratory. It's this incredibly diverse group of single-celled eukaryotes, most of them living in water. They are the absolute essential link between the simple bacteria we just talked about and the three big kingdoms we're familiar with: plants, animals, and fungi. And their lifestyles are all over the place. Some make their own food like plants, some hunt like animals, and some can even switch between the two. Because this kingdom is such a wild melting pot, when you look at its major groups, it's almost like you're seeing the first drafts, the early prototypes for the more complex kingdoms that were still to come. And you can see it laid out right here. You've got plant-like protists, the chrysophytes and dinoflagellates that were mastering photosynthesis. You have the slime molds, which were behaving a lot like primitive fungi, decomposing dead stuff. And then you have the protozoans, which were acting like the very first animals, hunting, moving, and eating other things. Okay, first up, let's take a closer look at the groups that were basically the primitive relatives of plants. We'll start with the chrysophytes, a group that includes some of the most important organisms in our oceans. This group is really all about the diatoms. These things are incredible. They live inside these intricate little cell walls made of silica, basically tiny glass houses. They're so incredibly abundant and so good at photosynthesis that we call them the chief producers of the ocean. And over millions of years, as they die, their indestructible little shells pile up on the ocean floor, forming what we call diatomaceous earth, which we actually use for all sorts of things, from polishing to filtration. Now, our next plant-like group, the dinoflagellates, are known for being, well, a little more dramatic. They're mostly marine, they're photosynthetic, and their cell walls are made of these stiff cellulose plates. And their most defining feature really comes down to this number right here, 2. Dinoflagellates have two flagella, those little whip-like tails. One runs down their length, and the other sits in a groove around their middle. This setup makes them spin like a little top as they move through the water. When conditions are just right, some dinoflagellates, like one called Goniolax, can multiply so fast that they actually turn the ocean red. These red tides can release powerful toxins that are really harmful to other marine life. And some can even produce their own light through bioluminescence, which just goes to show you how dynamic this group is. So now we get to the groups where the lines really start to blur. Take euglenoids. They are the ultimate hybrid. If there's sunlight, they act like a plant and photosynthesize. But if it's dark, they switch gears and become predators. Then you've got slime molds, which behave like primitive fungi. When things are good, they can swarm together to form this huge mass called a plasmodium, which then goes on to produce spores. And that brings us, finally, to the protozoans. These are widely considered to be the primitive relatives of the entire animal kingdom. They are all heterotrophs, which means they have to hunt or scavenge for their food, just like animals do. So how do we tell them apart? Well, we group them based on a very animal-like trait: how they move. You've got the amoeboids, which use these false feet to kind of ooze and creep along. Then there are the flagellates, which are propelled by a long whip-like flagellum. The ciliates are covered in thousands of tiny little hairs that beat together like oars, and they often use them to sweep food into a little mouth-like cavity. And finally, you have the sporozoans, which don't really move at all as adults. They're parasites with a life stage designed purely for infection. And while a lot of protozoans are harmless, this group unfortunately includes some of humanity's most serious pathogens. We're talking about entamoeba, which causes dysentery, trypanosoma, which causes sleeping sickness, and of course Plasmodium, the sporozoan parasite responsible for malaria. Their impact on human health has been absolutely immense. Okay, that was a whirlwind tour through the microscopic origins of complex life. So, what are the absolute most important high-yield points you should walk away with? Let's nail them down. So let's tie it all together. You've got the rule-breaking mycoplasma, the only prokaryote with no cell wall. You have the glassy diatoms, whose remains literally build up our ocean floors. It all funnels through the kingdom Protista, that critical connecting link that experimented with all these new designs, which gave us the protozoans, the first drafts of animal life, all sorted out by the very way they moved.
It really makes you wonder, doesn't it? The incredible journey from a simple prokaryote to the vast complexity we see today was pioneered in this microscopic world, and it just leaves you thinking, if these were life's first drafts, what other incredible designs and evolutionary secrets are still hidden?
All right, let's dive in. Today, we're going to unpack the incredible story of how scientists took on one of biology's biggest puzzles: how to create a map for all of life on Earth. I mean, with millions and millions of species, the task was just monumental. So where do you even start? Seriously, just stop and think about that for a second. Our planet is just bursting with life. You've got bacteria that can live in boiling hot springs, and then you have the blue whale, the biggest animal that has ever existed. How in the world do you start to make sense of all that beautiful, wild chaos? Well, you need a system. You've got to find a way to group things based on what they have in common, and, how they're related in the grand scheme of things. The very first attempts at classifying life weren't really about science at all. They were about survival. Early humans grouped things based on pure practicality. Is it something I can eat? Can I build a house with it? Can I make clothes out of it? It was a logical start, for sure, but it didn't really tell us anything deep about the organisms themselves. Then came one of the first big scientific tries from the ancient Greek philosopher Aristotle. He used things he could see with his own eyes. For plants, he was like, okay, are they small like herbs, medium like shrubs, or big like trees? Simple enough. For animals, he'd group them by whether they had red blood or not. It was a step in the right direction, but it was just incomplete. It left out this huge, invisible world of microscopic life that didn't fit anywhere. Fast forward a few centuries, and a botanist named Carolus Linnaeus steps onto the scene. He comes up with this really bold and, for its time, kind of brilliant system designed to classify every single living thing that was known. So Linnaeus' solution was to split all of life into just two kingdoms, Plantae and Animalia. The rule was super straightforward. If it has a cell wall and makes its own food, boom, it's a plant. If it moves around and has to eat other things, well, it's an animal. And for a while, this brought a whole new feeling of order to the world of biology. But you can probably see where this is going. That simplicity was also its biggest problem. It's like trying to organize a massive library by just sorting books into hardcover and paperback. Sure, it's organized, but it's not very useful, right? You'd have a cookbook right next to a horror novel. The system misses all the important connections and what the content is actually about. And as our scientific tools got better, especially with the invention of the microscope, those little cracks in the two-kingdom system started to look like massive canyons. We were suddenly discovering this whole new universe of life that just broke all the rules. Now, this slide is absolutely crucial because it shows the three fatal flaws of that old system. First, and this is a big one, it didn't separate the super simple prokaryotic cells, like bacteria, from the complex eukaryotic ones. Second, It just threw single-celled organisms in with giant multicellular ones. Think about something like Euglena, which can swim and photosynthesize. It just didn't fit. And third, it incorrectly lumped things like fungi, which don't make their own food and have a different kind of cell wall, right in with the plants. It was, to put it lightly, a total mess.
So this mess needed a totally new solution, a revolution, really. And that revolution came in 1969 from an ecologist named R.H. Whitaker. He came up with a new system of classification that would completely change how we look at the tree of life.
Whitaker's genius was that he didn't just use one simple rule. Nope. He built his system on 5 much deeper criteria. He looked at cell structure. Are we talking prokaryotic or eukaryotic? He looked at body organization. Is it one cell or many? But the real game changer was making the mode of nutrition, how it gets energy, a central pillar of the system. And for the first time, he officially included phylogenetic relationships, which is basically the evolutionary family tree that connects all life.
And here's the beautiful result. 5 clean, distinct kingdoms. Monera became the exclusive home for all the simple prokaryotes. Protista was created as a sort of catch-all kingdom for the diverse single-celled eukaryotes. Fungi, finally, got their own kingdom, defined by their chitin cell walls, and that left Plantae for the multicellular photosynthesizers and Animalia for the multicellular heterotrophs that eat their food. Suddenly, everything just clicked into place. This new map of life was so much more accurate. All those organisms like bacteria and fungi that had been shoved into the wrong boxes now had a proper home, one that actually reflected their true biology.
Okay, so to see just how powerful this system really is, let's put it to the test. Let's zoom in on that first, most fundamental kingdom and see how Whitaker's rules bring order to the massive world of bacteria. Kingdom Monera. Every single member of Kingdom Monera shares this one defining feature. They are all prokaryotes. What does that mean? Well, it means their cells are incredibly simple. There's no true nucleus, their genetic material, their DNA just kind of floats around freely inside the cell.
And here's the key takeaway for this kingdom, a really important point to remember. Monera is only for bacteria. They are the sole members. These little guys are the most ancient, most widespread, and most metabolically diverse life forms on the entire planet.
But even inside this one kingdom, there's a ton of diversity. So the system helps us break it down even further into 2 big groups. On one side, you have the archaeobacteria, the ancient bacteria that live in some of the most extreme, hostile places you can imagine. And on the other side, you have the eubacteria, or true bacteria. These are the ones you find pretty much everywhere else, and they have that classic rigid cell wall made of something called peptidoglycan. And when we say extreme, we mean extreme. You've got halophiles that love super salty water. You have thermoacidophiles that are just chilling in boiling hot springs. And then there are methanogens, which pump out methane gas and live in swamps and even inside the guts of cows. These things are the ultimate survivors. The eubacteria, on the other hand, are famous for having the most incredible nutritional diversity. Most of them are heterotrophs, breaking down organic stuff for food. But then you've got some, like cyanobacteria, that are photosynthetic. They use sunlight for energy just like plants do. And then you have others that are chemosynthetic, getting their energy from chemical reactions. Just an amazing range of strategies for survival. And that right there is the beauty of Whitaker's system. It takes this unimaginably huge and complicated world of microscopic life and just organizes it with a logic and a clarity that the old 2-kingdom model could never, ever achieve. And this brings us to a really, really important idea. Biological classification, it's not about making a final fixed set of rules. It's a living, breathing science. It's really a story that we are constantly updating as our technology gets better and our understanding of life's history gets deeper. So that leaves us with one last really cool thought to chew on. We went from Aristotle's simple groups to Linnaeus's 2 kingdoms, and then to Whitaker's 5. But guess what? The story doesn't stop there. Today, because of genetic sequencing, many scientists have moved on to a 3-domain system that actually separates the archaeobacteria into their own domain, on par with all animals, plants, and fungi combined. It just goes to show you, the map of life is all. Welcome back. Today, we are digging into one of the most fascinating, absolutely vital, and probably most misunderstood kingdoms on the entire planet, the world of fungi. So let me just start with a quick question for you. What could a mushroom popping up in a forest, the fuzzy mold on a piece of old bread, and the microscopic yeast that gives us beer, what could they possibly have in common? I mean, they seem totally different, right? Well, it turns out they all belong to one single incredible kingdom. And to really get our heads around fungi, we first have to understand that they are completely in a league of their own. Seriously, they aren't plants, and they are definitely not animals. They are so unique, they get their very own exclusive club, Kingdom Fungi.
Okay, and this is where it gets really strange. You know how plant cell walls are made of cellulose, right? Well, fungi build their cell walls out of something called chitin. And get this, that is the exact same tough stuff you find in the exoskeletons of insects and crabs. Plus, unlike plants, they can't make their own food through photosynthesis. Nope, they have to go out and find it, just like we do.
So, what is a fungus actually made of? Its body isn't roots or stems or leaves. Instead, it's built from these super long, incredibly thin threads called hyphae. And when all these threads weave together, they form this huge, interconnected network called a mycelium. You can think of it like a massive hidden underground internet. And the mushrooms we see, they're just the tiny part that pops up above ground.
Right, so if they can't make their own food, how in the world do they eat? Well, this is where fungi become absolutely essential for all life on Earth. Their feeding strategy really defines their role in the ecosystem, and it basically breaks down into three main approaches.
And these three lifestyles are everything. First up, you've got the saprophytes. Think of them as nature's ultimate cleanup crew. They are the decomposers, breaking down dead stuff like fallen leaves and logs. Without them, we'd literally be buried. Then you have the parasites, which live on and feed on living organisms. And finally, my favorite, the symbiotics, which team up with other organisms to form these incredible, mutually beneficial partnerships. And these symbiotic partnerships are just amazing. Take lichens, for example. You see them on rocks and trees all the time. That's not one organism. It's a team up between a fungus and an alga. The fungus provides a safe house, and the alga makes the food. Perfect. Or look at mycorrhiza, where fungi wrap around plant roots, acting like a giant extension cord, pulling in water and nutrients for the plant in exchange for a little sugar. It is the perfect win-win. So this brings us to a huge puzzle. Fungi are everywhere, from tiny single-celled yeasts to massive underground networks that span for miles. They have all these different ways of eating and living. So with all this mind-boggling diversity, how on earth do scientists bring any kind of order to this kingdom? Let me put the question to you. How would you even begin to classify organisms that are this varied? Do you sort them by what they look like, by how they eat, by where they live? It seems almost impossible to find one consistent rule that works for all of them. But there is one. Believe it or not, it all comes down to how they reproduce. Yup, specifically their sexual cycle. The secret to unlocking the entire fungal family tree, the key to it all, lies in the unique ways different fungi create and release their sexual spores. So how does that even work? Well, it's kind of like a three-step dance. First, two compatible fungal cells merge their bodies together in a step called plasmogamy. Then, in a really crucial second step called karyogamy, their genetic nuclei actually fuse into one. This new combined cell then undergoes meiosis, which is just a special type of cell division to create a whole new batch of genetically unique spores, all ready to start the next generation. And that right there is the key to everything. The specific way a fungus produces its sexual spores, the kind of structure it builds, and the type of spores it releases is the primary characteristic scientists use for classification. You can think of it like a unique reproductive fingerprint for each fungal family. So, now that we have the key, let's go ahead and unlock this puzzle. Based on their sexual reproduction, fungi are sorted into 4 major classes. So, let's meet the families. And this is how it all breaks down. You've got the phycomycetes, or algal fungi, then the ascomycetes, known as the sac fungi, which includes famous ones like penicillium, and the basidiomycetes, or club fungi, which includes the common mushrooms you know. But look at that last column, the sexual spores. Each family is defined by its own unique type, except for that last group, the deuteromycetes. For them, it's a complete mystery. And you can really see how different those spores are. If you look at the ascomycetes, the sac fungi, they typically produce their spores, called ascospores, in this neat little packet of 8 inside a special sac. But the basidiomycetes, the club fungi like mushrooms,
they produce just 4 spores on the outside of a tiny club-like structure. It's these tiny structural differences that define everything.
Okay, so now we have to talk about that last mysterious group. They represent this really fascinating little quirk in the world of scientific classification. They are known as the imperfect fungi. So why call them imperfect? It's not a judgment. It's not like they did anything wrong. In the world of mycology, the perfect stage of a fungus just refers to its sexual reproductive stage. So the deuteromycetes are basically a holding category, a kind of scientific waiting room for all the fungi where scientists simply haven't yet observed or discovered how they reproduce sexually. We only know their asexual or imperfect stage.
But, and this is a huge but, there is one major clue that links these mystery fungi to another group. Just one common thread. While their sexual stages are a total unknown, their asexual spores, which are called conidia, look identical to the asexual spores produced by the ascomycetes. This is a massive hint that suggests many of these so-called imperfect fungi are actually just sac fungi that have either lost or are just really, really good at hiding their sexual stage.
And that brings us to our final thought for today. Given that really strong link in their asexual reproduction, if a scientist finally observes the sexual or perfect stage of a fungus that's currently sitting in the deuteromycetes group, which of the other families would it most likely be moved to? It's a great...
All right, let's get into it. Today, we're going to venture to the very, very edge of biology. We're talking about a world filled with the strangest partnerships you can imagine, microscopic hijackers, and even things that make us question what it even means to be alive. You know, we usually think of nature as this world of competition, right? Predator and prey. But it's also packed with these incredible examples of teamwork. But what happens when that teamwork gets twisted? Or what happens when the thing attacking isn't even technically alive? That's the wild ride we're taking today.
So first up, let's kick things off by looking at some of the most successful and kind of weird partnerships out there, where 2 totally different organisms realized, you know what, we're just better together.
Okay, meet the lichen. Now, you've probably seen this stuff growing on rocks or trees, and you might think it's just some kind of moss. But it's not one thing at all. It's actually a team, a fungus and an alga living together in this permanent symbiotic mashup. It's basically 2 different life forms that have fused to become one.
So how does this team up actually work? Well, it's a pretty sweet deal. The fungus acts like the house. It provides the structure, it soaks up water, and it grabs minerals from the environment. And in return, the alga, which can do photosynthesis, acts as the live-in chef, cooking up food from sunlight for both of them. It's an absolutely perfect arrangement.
And because they're such a great team, lichens can do some incredible things. They're often the very first form of life to show up on bare rock, slowly breaking it down and creating soil so other plants can eventually grow. Oh, and here's a cool fact. They are super sensitive to air quality, so if you see a bunch of lichens around, chances are you're breathing some pretty clean air. If they're gone, that's a bit of a warning sign. And this idea of a fungus partnering up, it's not a one-off thing. Take mycorrhiza. This is another team-up, this time between a fungus and the roots of a plant, like a big pine tree. The fungus basically acts as an extension of the plant's root system, massively boosting its ability to suck up water and nutrients. And what does the fungus get? The plant sends down sugars to keep it fed. It's like a massive underground internet for nutrients, and it's absolutely essential for a healthy forest.
But of course, not all of these close relationships are so win-win. So let's shift gears from helpful partners to the ultimate freeloaders, the parasites. We are now entering the bizarre world of viruses. These are biology's great hijackers, and they live right on that fuzzy line between living and non-living.
So here's the big question, the paradox at the heart of it all. Are viruses even alive? Well, it's kind of yes and no. When a virus is just floating around outside of a living cell, it's completely inert. It's no more alive than a grain of sand, just a lifeless little crystal. But the second it gets inside a host cell, everything changes. It springs to life, it starts replicating, it evolves. It's this two-faced nature that makes them so fascinating and honestly, so dangerous.
And if you look at their structure, they are ridiculously simple. A virus is not a cell. It has no nucleus, no metabolism, nothing like that. It's literally just a little snippet of genetic instructions, either DNA or RNA, that's been packaged inside a protective protein box called a capsid. Think of it like a microscopic syringe filled with code, and its only purpose is to inject that code.
And get this, even bacteria, some of the tiniest living things on the planet, have their own viruses that hunt them. They're called bacteriophages, and they look like something straight out of a sci-fi movie. These things are hyper-specialized little predators that hunt down and infect only bacteria. It just goes to show, no matter how small you are, there's always something smaller ready to get you.
You thought a virus was simple? Ha! Prepare to go even simpler. We are now diving into a realm of infectious agents that make viruses look downright complicated.
The year is 1971. A scientist named T.O. Diner is studying a potato disease, and he stumbles onto something that just completely breaks all the rules. He found an infectious agent that was even smaller, even simpler than any virus known to science.
He called this thing a viroid, and the difference between it and a virus is just stunning. Remember, a virus is genetic code inside a protein box. A viroid, it's just the code. That's it. A tiny, naked loop of RNA with no protective coat, no box, nothing. It is the absolute bare-bones minimum you need to be an infectious agent.
So, to break it down, a viroid is just a free-floating piece of infectious RNA. It's way smaller than any virus, it has no capsid, and yet it can get inside a plant cell and cause chaos, like the potato spindle tuber disease that Diner was studying. It's basically pure, weaponized information. Okay, so if naked RNA wasn't weird enough for you, get ready for prions. Because this isn't about genetic code at all. This is something else entirely. We're talking about an infectious agent made of nothing but protein. No DNA, no RNA. Just a protein that's folded into the wrong shape.
Here's the terrifyingly simple way it works. A prion, which is a misfolded protein, just bumps into a normal, healthy protein of the same type. When it does, it forces the normal protein to change its shape and become a misfolded prion too. It's a chain reaction, like a zombie protein converting others, creating a cascade that just destroys tissue, especially in the brain. This is the horrifying mechanism behind diseases like mad cow disease and its human version, Creutzfeldt-Jakob disease.
Okay, wow, that was a lot of strange biology, right? We've gone from partners to parasites to rogue proteins. So to help you get all these mind-bending ideas straight, let's lock in some key facts and put everything in its place. Think of this as your study guide.
Okay, so this table is super useful for seeing the big picture. Just look at the first row, cell type. You see that fundamental split between the prokaryotes, like bacteria, which are simple cells with no nucleus, and the eukaryotes, like fungi, which are the more complex cells that do have one. These basic differences are how biologists start to sort out all the incredible diversity of life.
All right, need a super fast way to remember the 5 kingdoms of life? Just remember this sentence. My poor friend practices algebra. That's for Monera, Protista, fungi, Plantae, and Animalia. Easy, right?
And what about the main classes of fungi? We've got one for that too. Just think, please accept bread daily. That'll help you lock in Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes.
And finally, let's do a rapid-fire cheat sheet of some must-know facts. Ready? Smallest living cell, mycoplasma. Smallest infectious agent, that's the viroid. A non-living infectious agent, the prion. The first species to colonize bare rock, our buddies the lichens. Those fungi that we call imperfect because we haven't found their sexual stage, that's Deuteromycetes. And those infamous red tides, they're caused by dinoflagellates. So, we end right back where we started, on that blurry, fascinating line. A prion is just a chemical, a molecule of protein. It's not alive, it has no genes, and yet it replicates by forcing other chemicals to copy its corrupted shape. It completely blurs the line between a simple chemical reaction and a biological infection. Which really makes you wonder, where does the chemistry of a molecule